Good morning. So my name is Nick Carroll. I'm Associate Professor of Chemical Engineering at the University of New Mexico. As you can tell from the title of the presentation, this is my first foray out of the safe academic bubble. So let's just rename the title of this talk, Radioactive Silica Spheres for Personalized Treatment of Liver Tumors. Okay, so I think that's a little better, right? Let's see, how do we change this? Okay, so actually what I will do, which silica sphere treatment of liver cancers. So hepatocellular carcinoma, or HCC, accounts for the vast majority of liver cancer cases and nearly 1 million deaths each year worldwide. So in addition, 50% of colorectal cancers metastasizes to the liver, and the 5-year relative survival rate is 22%, right? And this is a big problem, particularly in New Mexico, which is really, a hotspot for this type of cancer, particularly with the Hispanic and Native American population. So one treatment that has gained popularity, because this is a localized treatment, is to inject highly radioactive microspheres into the tumor space. And they typically, of course, clinicians want to do this at a high radiation dose, right? And part of the problem with delivering high radiation dose and large amount of the therapeutic to the local tumor space is that, quite frankly, clinicians and doctors, we just don't know where these beads are accumulating, right, in the body. And one thing that we want to avoid is these highly radioactive spheres damaging healthy tissue. Okay, so right now the state of the art is, folks are using resin spheres and other types of materials, radioactive materials, to deliver the therapeutic to the tumor space. However, these dosages are kept artificially low to avoid damage to the healthy tissues. And so one thing that we need to do is to understand where these spheres are going. This is exactly what the focus of this project is. So I think, we've talked about, so where the market stands now is there are 2 vendors who basically dominate the market for produce these beads very quickly. And in addition to this, the fabrication process it involves very large reactors. Okay, so to irradiate these spheres and so forth, there are just a few neutron reactors that are available. There are 5 available worldwide, and so getting time on these reactors and so forth is problematic. Okay, so actually, I don't know if I can go back. So what we have done, the innovation that we have developed at UNM, is to create a microchip which can fabricate these glass radioactive beads. And so we developed a method to rapidly manufacture these glass microspheres in just a few hours. And the idea is that we can have this, let's say the morning of some type of treatment, we can generate these particles essentially bedside. As I mentioned, the current state of the art involves a neutron activation of glass microspheres in one of these, only one of 5 high-flux nuclear reactors worldwide. These radioactive glass microspheres that we're making can be used in the rapidly expanding field of theranostics, which is not just therapeutics, but also diagnosing real-time the accumulation of these beads. And as I've mentioned, radioactive microspheres are currently used to treat liver cancer, but this is suboptimal. So without going over too many of the details of the engineering, we've developed essentially a lab-on-a-chip process that greatly miniaturizes what we're doing. And so this is a prototype chip which I brought. So instead of having a huge industrial reactor and so forth, we're able to fabricate the idea behind that is we fabricate these silica spheres using reagents that are can be fabricated in any radiology lab. And so that equipment already exists. And the idea is we take these precursors, we run them through the chip, and this is a very fast and efficient way to create these microchips. And so the idea then behind these theranostics that we have in terms of commercialization is in fact, to create a benchtop fabrication sphere model where essentially a clinician or a technician under a clinician's supervision, let's say, the morning of some type of, whether it's therapeutic or whether it's diagnostics, in terms of imaging with these microspheres where they are accumulating for each patient, is you can dial in the size of the beads, the dosage, the radioactive dosage that you would like. And then, the idea is to build this benchtop autonomous sphere fabrication unit. So this is what we're working on now is automation of the process. So we're using artificial intelligence, machine learning, to have an autonomous operating device. And then the next thing that we would like to do then is to build these benchtop fabrication devices. And one thing that could be a continuous business model is to develop inserts, microfluidic inserts and fabrication inserts that would go into these machines, as well as potentially cartridge units that have the precursors and so forth. And so this is the general idea. So data and results. So right now we have developed prototype microchips. We specifically gallium, fluorine, and copper. What this table shows is that really a better heading for that first column would be a predicted activity. So this is the radioactivity. You see that the product activity matches very closely. In other words, the radioactive activity of these silica microspheres. And then the waste activity is the amount of radioactive material that would leak or be unwanted leakage of that material from these beads. And we see that that is very low. And so just to go back, so the team comprises Reitz Owen, who is from radiology. So he's doing most of the biomedical engineering aspects of this work. Andy Shreve, who is an expert in automation and process control and artificial intelligence machine learning. So he would be helping with the automation. Gabriel Lopez, who's an expert in biosensing and materials, and Ian Larson, who is the PhD student who did most of the work in this on this project, most of the experimental work. Okay, so that's basically this very early idea that we have in terms of commercialization of these imaging and eventually therapeutic radioactive microspheres, and I'll be happy to take your questions.
Great presentation, Nick, and good job on your first foray out of the lab.
Thank you.
Question on who's going to be the team member who understands how to commercialize this? Do you have somebody who's not a doctor or scientist?
No, right now we don't have anyone like that on the team, and I think right now we're relying heavily on Rainforest at UNM, which is an IP development, organizations pale in comparison to the Rainforest. It's one of the best in the country. So right now, I'm working with Alex Rorick, who's a very positive influence on removing me from my academic bubble, as it were, and moving it, more into the commercialization space. But this is something that we need to address, I think. Yeah.
It seems to me, great presentation, Nick. It seems to me there's a couple areas that you have to move in parallel. One is sort of the clinical side. How, on a scale from 1 to 10, how far along are you on, say, being in a place where you're ready to start doing clinical trials? And on the product hardware side, you showed a concept of a product. What's required to get that built, a prototype?
Oh, thank you very much for these questions. So in terms of deployment to the clinical trials, I would say this is somewhere where, doing this research in an academic setting is an advantage because we have the medical school. And Reese Owen is already doing clinical work with resin beads that are these commercially available beads. And so this is something that we're working actively on. The next step is to write an NIH grant, which will allow us to first deploy these beads in mice models. And then, the next clear evolution from that is to work with Reed on actual clinical trials. And then I think, in terms of making this benchtop device for on-demand synthesis of these therapeutic beads, I could envision, maybe some type of privatization and maybe an SBIR grant to get started with, actually building prototypes of these, benchtop devices to create these radiospheres.
Anyways, coming back to the question, there are some novel ideas, amazing ideas, but they got lost in translation because of the convoluted world of healthcare system that we have to deal with. Do you foresee any immediate term engagements with any provider network and maybe payer network as well, because you need to take care of all those blocks ahead in time rather than kind of trying to fix them along the way? So have you thought about it? Is it too early to discuss that?
Right. No, in fact, we haven't thought about that, but but I appreciate you bringing that up because now that's something that I think we definitely should take into consideration. And again, that's, it was a privilege to present at Innovate New Mexico just for this, because that's not something I was considering in the next evolution. Yeah.
No, thank you for such of the context. It's really exciting to kind of hear like what's being built, and congrats on stepping outside of the comfort zone. It's really exciting to see people taking the courage and the leap of faith to do this. So I appreciate that.
Thank you.
I, as the last kind of person asking the question, as I've like really thought about this and kind of started toying about it, and maybe this is just more of like a recommendation of kind of thinking about what does a product roadmap look like, and how do you start identifying, early revenue opportunities as you start building out this roadmap?
I really appreciate that, and I think just even being up here and having these really engaging questions, your mind starts to operate like initially perhaps we could utilize our academic connections to other medical schools and other places where eventually perhaps we could provide them with free prototypes that could then be about. Okay, thank you.